And so without further ado, please join me in giving a warm welcome to our first keynote speaker, Professor Tony Mok, Chairman of the Department of Clinical Oncology of the Chinese University of Hong Kong, presenting Redefining Cancer Care, Smarter Therapies, Global Impact. Professor Mok, please take a seat. Can everyone please be seated?
Hi, thank you so much. It must be more fun to chat than listening to me talking, I guess. So I will just wait one moment for the gentleman in the back there to sit down before we start this talk. Well, first of all, it's my great honor to be able to be the first speaker in the New Frontiers International Conference. Actually, it's most exciting to be part of this big family, and I can see that the significant collaboration between Hong Kong and the Great Bay Area and China. I just have to wait for the back to be a bit quiet down before I can start talking. Right, thank you so much, thank you. So I actually had the joy of talking about Redefining Cancer Care and any smarter therapy. So this is my disclosure, and more specifically, I'm on the board of directors of these three companies, AstraZeneca, HUTCHMED, and Insight. Now, I want to say that cancer care is a huge topic. This is brief, is actually on the definition of cancer care by WHO. You can see it actually enclosed a number of things. Treatment is only part of it. There's a multidisciplinary team. There's also of the patient choice, as well as the quality of life and patient journey. So it's a huge topic that unlikely I'm going to touch in great detail today. But more so is the fact that it's a hugely expensive journey. And 2020, this is from United States, just looking at the top cancers, they actually spent about $209 billion on cancer care alone. So with such a big topic, I'm actually going to touch on about the smarter drug platform that we're looking in the future, the smarter biomarker, which we can select the patient, actually with actually more insight on who may benefit most from the treatment. And last but not the least, with the last few minutes to touch on a little bit on the global impact. Now, smarter drug platform, let me just clarify the one fact. We actually have been using a smart drug in the past. Molecular target therapy, immunotherapy, all this can significantly improve the overall survival of the patient. I just cite a few examples, Tagrisso, lorlatinib, and also pembrolizumab on actual treatment of lung cancer. According to genomic selection, we actually make great impact, and all this turn into $1 billion drugs. Very significant impact on the economy as well. So the question is that, are we making smarter or just more novel? Now, we have to be very careful. Something new, a better concept, doesn't always mean that it's going to be smarter or better. We have to prove it. I named actually about 6 of them: antibody drug conjugate, bispecific antibody, radionucleotide, PROTAC, cancer vaccine, cell therapy. All this actually already had multi-billion-dollar investment in it. Obviously, limited by time, I'm not going to touch each one in detail, but I'm going to touch on the concept, where we are, and what are the major problems in very brief fashion. Antibody drug conjugate. We use an antibody to target the cancer antigen, and then tacking onto the antibody, we have the cytotoxic drug. So in a way, it's a zoom in into the cell and kill the cancer cell. Very sound concept, have been around for a long time. And the first, there's the fact that multiple companies in China, internationally, already pour in multi-billion-dollar in the investment. And you can see that major target include HER2, TROP2, MEK, AXL, EGFR, a very lengthy list of targets that we may have. Now, the most successful one is actually Enhertu, which actually AstraZeneca we bought for $6.3 billion from Daiichi Sankyo, but now we're actually generating billions of dollars every year from selling this drug for patients with actually breast cancer and lung cancer. But this is a successful story. But there are also stories that are not so successful. Anti-TROP2, with this drug called deruxtecan, we actually had a positive study, but marginally positive, better than chemotherapy. Response rate is barely double, almost 20-some%, not hugely successful. So we need to make actually better because there's so many on the so-called the bar right now that we have to leap over to make this successful. So definitely, we need a biomarker. And so for that, I'm going to talk a little bit more about biomarker in the future. Without biomarker selection to know which patient benefit from this type of drug, I think we're going to face failure. So for our development, it's not just the drug, but how to select the patient with a sound and smart biomarker. Bispecific antibody. We now have the technology to create all kinds of bispecific antibody. And the list is actually tremendously long. Now, because we're successful in the PD-L1 for immunotherapy, so now the combined is PD-L1 and CTLA-4. Both actually are the target that won Nobel Prize. So now bispecific antibody, one of the three drugs is actually 5752 from MacImmune. And then actually the first study was demonstrated that originally, patients with high PD-L1, with the PD-L1 inhibitor, we have the result. But with the bispecific, as you can see, the red are the one with the less than 1%. Without the PD-L1 expression, the bispecific also got evidence of improvement. But then there is actually randomized study ongoing. This is one of them, and we still have to wait and see. But for that, there's actually one China drug I would like to mention. This is a bispecific antibody that come actually, they're actually the so-called the primary office is actually in Zhongshan in the Greater Bay Area, Akeso. And so they make a drug called avulisumab. It's bispecific of anti-VEGFR and PD-L1. And they did a randomized study against a standard international drug, pembrolizumab from MSD. And this is the first time they were able to say that PD-L1 above 1%, but then randomize about these two drugs and to see which one's better. But there's a slight trick on this study. Because 1 to 49, actually the standard should be chemo plus immunotherapy. Over 50%, it should be immunotherapy alone. So there's a slight trick with this study, but they actually had a tremendous success. For that, I actually share a little bit more. It become on stock market, tremendous success, with this Akeso drug. The number is 9926, just in case you want to know. Now, what do we need? Again, we have a bispecific, we need a dual biomarker. So again, go back to the concept of a biomarker that we have to invest on. Now, cancer vaccine is sound. In a sense, is that we stimulate, educate the immune system to recognize the cancer to cure it. And we have actually a lot of investment. One of the biggest companies is BioNTech, the one that produced mRNA for COVID. They make a lot of money from the COVID, but now they go back to look at the cancer vaccine investment. So one of the drugs is called BN-116. And then this drug actually already in clinical stage, and then looked in multiple clinical trials. And the early data, actually just come out, phase 1 data, was actually quite impressive, suggesting the potential of efficacy. But there's one problem, is that not one vaccine can be fit for every patient. So the other important concept is so-called personalized vaccine. We take out your tumor, do your DNA analysis, find out which is the most likely antigen, create a vaccine for you. So the personalized vaccine is actually about 3 or 4 companies globally are actively engaged in this. But if you want to invest in this area, may I just warn you about one thing. Cancer vaccine doesn't work fast. You get the vaccine, you educate the system to create your immune system to fight the cancer, but it does not work immediately. So if you would put an end-stage patient, the time required for this to work may not be sufficient for the patient to survive. So cancer vaccine, barrier, we need the time. We have to find the right patient, have sufficient time for the cancer vaccine to work. Now, there are all kinds of cell therapy this day. Some are true, some are not true. One of the actual hot topic is the tumor-infiltrate lymphocyte, meaning that we identify the white cell already infiltrated in the cancer, take them out, grow them, and give it back to the patient. And this is actually one study looking at the tumor-infiltrate lymphocyte. So it is actually a very complex process. We have to harvest the tumor, and then we have to expand the T cell, and then we have to prepare the patient with some chemotherapy, and then we infuse it back. So in this particular study, we start off about 32 patients, only 14 patients end up having the drug. So, but then we are able to see response. So this is actually one of the first early study demonstrating the tilt is actually effective. Now, there are a lot of other CAR-T, but I will not spend the time to talk about it because of the limit by the time allotted to this talk. But then, what we need is scalability. You can see that the procedure itself is so complex. You can do it in academic center, but make it scalable is very difficult. So we have to find smarter way. How can we manufacture TIL into a clinical situation? Now, biomarker platform. Now, we actually always need a biomarker to select the patient. This is an ideal biomarker. The red guy are the one that respond, and then we use a biomarker, select them, 100%, high sensitivity and specific. That's perfect. But most of the time, we have not so smart biomarker. We want to select the red guy, but we only select part of the red guy, and then we get some yellow guy into it and thought that is the red guy. And as a result, they do not work. The response rate becomes low. So how can we make a smarter biomarker? Now, as you can see, the ADC is a huge market. Then what is the biomarker? Well, of course, we try to target the protein. And that may be the one because they attack the protein get it into the cell. As a result, it may work. But unfortunately, most of the biomarker, not just looking at the protein, it doesn't work so well. This is another biomarker for HER3, and then the biomarker is totally useless. Now, why is that? Well, one of the reasons is that we are actually looking at things in a different way. Now, first is that how detailed we look into it. So transforming it in the TROP2 situation, they actually don't have a biomarker. But this is actually something new called the quantitative continuous score. They use AI to look at each protein and quantify them inside the cell and outside the cell. As a result, they create a ratio and then identify 75% of patients with this ratio was considered to be positive. And based on their randomized study, they were able to identify the fact that, hey, squamous cell, they don't actually have such a good expression. Other cells, they may have actually better expression. So as a result, they were able to demonstrate that the patient with a positive QCS score were able to have a better result compared to a low one. So in a way, is that the next wave of the biomarker is going to be quantification by AI technology. That's another dimension, is the fact that we actually had always looked in tumor in a two-dimension fashion with a pathological slide. But nowadays, actually in CUHK, we have developed three-dimensional histopathology. So this is actually very interesting in a sense is that because we can look at tumor block, and then we can quantify a three-dimensional tumor block such as I showed you. Just like what happened when you low down. So in the surface, when you look at the protein, it just gets the soy sauce to the surface, but it doesn't taste so good. For good low down, you've got the soy sauce to go inside. So what is the technology to bring the soy sauce inside the egg? Now we have actually this technology in CUHK, and which is actually using a special compound. We only disclose part of it to you. It's called cyclodextrin. And then we use the cyclodextrin to bring in the antibody, and in the same time, we can bring in multiple antibody. Just show you one example that what we have done. This is actually a three-dimensional picture of a colon. The red is the vessel, the whitish is the nerve. As you can see that we can actually get a piece of colon and look at the three-dimensional fraction. Now, how does it actually apply to biomarker? Well, what we can do is that this is a tumor tissue. DAPR is actually the neutral one. TROP2 is the one antibody direction. HER3 is another one. So we can actually stain the tumor in a three-dimensional manner. As a result, we can quantify the tumor in a three-dimensional fashion. Let me just show you one other thing, is the fact that if you just look at the expression on the two-dimensional slide, if you take it horizontally, you will see that this slide will be actually TROP2 HER3 positive. But then if you take it slightly further down, it may become negative. You take it further down, it becomes partly positive. So depending on how you cut the tumor, randomly, you get positive or negative actually by default. So only 3D is actually going to give you the absolute quantification. So I think this is one future direction, is 3D biomarker technology. So moving on, I only get 1 minute and 21 seconds left, is the global impact. Now, I want to show you this graph, which actually come out to 2021 in the ESCO book. It's actually look at the cancer drug cost versus the GDP of the country. Now, in India and China, as you can see, are almost like 1/3 of the world population. The cancer dollar that we spend is not that high compared to the United States. But even though we're lower, if you look at in the percentage of the GDP, is thousands % above, meaning that most of the people in the general population cannot afford cancer therapy. So the question that we have is that how can a drug that's made in made in China, have an impact globally? Let me just cite you some example. This is actually the development of China briefly in a recent article from Nature in 2024. We classify the China-made drug as a me too, meaning that we just copy what other people have. But there's also the fast follower, meaning that we make improvement of the drug and then develop it faster. And then the first in class, meaning that we modify it to make it the best in the class. Now, if you look into China's situation, we actually, in 4 years' time, we actually double our first-in-class number of drugs. Right now, we had over 400 first-in-class drugs actually under development in China right at this moment. And you can see the number of drugs in total also increased tremendously over the years. So the question is that, how could this drug benefit the world? So, first of all, the targets are similar. That's actually not to innovation at this point, but we can make it better. Just like, your sneaker, we do it for Nike, but then now the China make their own brand is actually even better than Nike. So, based on this concept, how do we benefit the world? So, one thing is that a drug got NMPA approval, but then if you really want to globalize, then you actually have to study outside China and then get FDA, EMA approval before you can globally recognize it. Or NMPA, you just stay in China alone. Now, staying in China alone, possibly most of the pharmaceutical companies may not be able to have significant financial gain because the cost of the drug in China is relatively low. So one way is the fact that they license out. They out-license it to another company in US or international company such that they can get the global recognition from FDA, EMA, and then globalize. So that's where the situation is right now. Let me just cite you an example. I showed you about ivonescimab, Akeso. They actually used $500 million, actually out-licensed it to Summit in the United States. HUTCHMED, which is actually a Hong Kong, Shanghai-based company, we got a drug called fruquintinib. We used $400 million, actually Takeda gave us $400 million and out-licensed Takeda and now become a global drug. But there's one problem. Let me just show you this number. It's actually quite astounding. It's the fact that the monthly cost in China is $1,150. In Takeda, once it's sold to the United States, it's $26,000. Well, they have to get their investment back for the investment plus the extra clinical trial. So once you had gone through this out-licensing process, the drug can no longer be economical to the patient. So what is actually one potential solution is that NMPA, now we have high-quality research and high-quality registration center. But can we use the NMPA, make an individual country approval, and then globalize, one country at a time? Well, I think Hong Kong is a good example. Hong Kong, now we have something called a 1+ mechanism of new drug approval. All it is has to be unmet medical need, orphan drug designation, and then local clinical data. With that, they gain the so-called the local registration in Hong Kong. Fruquintinib is one fine example. Then actually, it is a situation that we have actually gone through it within a relatively short time, and now even that, after arrival in Hong Kong, is only slightly higher cost than the rest of the world. So for that, we have 2 randomized studies, one in Hong Kong, China, one global. In the future, if you can escape the global one, then we've saved a lot of costs. Allow me to summarize. I think cancer care is a very complex and expensive in general. And then we have a number of smarter drugs right now. And also in the future, we look at AI for the biomarker as well as the 3D histology. And hopefully, our China drug is going to make great impact in the world because of the great development we have in the country. So last but not the least, I would like to say that smart cancer care has the smart from smart healthcare entrepreneurial, the two of them. Thank you so much. Thank you.